FungiDB, a tool for DNA methylation research in Phytophthora. Methylation plays a role in various functions within a cell. For example, cell differentiation. It's used as a defense mechanism in bacteria, so it could differentiate between itself and viruses. So restriction enzymes can come in and kill the cell or the virus. It also used in DNA repair pathways. It plays a role in cancer and also in regulation of expression. Currently, there is a broad standing opinion that oomycetes do not undergo methylation. This has been reinforced by various bisulfite sequencing experiments. I am not here to refute that. I am here to suggest that it is present in a small fraction of its genome, and that there have been more accurate methods of identifying methylation in organisms within the past few years. They have actually identified methylation in Drosophila on the X chromosome, and they believe it plays a role in dosage imprinting. In the past, one of the reasons why we believe oomycetes undergo methylation is we have exposed them to a cancer drug known as 5-AZA. And it pretty much looks like a cytosine. However, right here at this 5' position, they switch this carbon to a nitrogen, which means that this methyl group will not be able to attach because the nitrogen only wants to bind 3 times. So this will be incorporated randomly into a cell, and it will pretty much remove methylation. When we did this, we saw a couple of different things. One, we saw some weird swim patterns in zoospores, and we also saw differentiation between expression levels of virulence genes, which indicated to us that we should look into a potential gene candidate that would be responsible for causing methylation in the genome. The gene that we found in our lab was the DMAP1 gene. It is proven in the past that it can actually cause methylation on its own, and when I came in, this was handed to me already, so I didn't have to actually find the gene. Alright, so when I initially started this, VIPA gave me my gene and she said to find expression data. Luckily enough, the previous year I was fortunate enough to go to the bioinformatics workshop, and I had a good idea how to do this thanks to that. So I went back to my previous instructions that I saved from there, and I first started off with a blast search. So I took my gene and I did a protein search blast against all other oomycetes through FungiDB. And from that initial search, I ended up with 45 genes. Of those 45 genes, you can see that my particular gene of interest is right here at the top with a very good score and 0 error. Now, for the rest of this presentation, I will be also concentrating on P-capsicity, mostly because I have expression data that I'll be presenting later through Galaxy and through the genome browser at FungiDB. So, now that I found my initial group of orthologs that I believe are orthologs, I did a reciprocal blast search just to confirm that these are in fact orthologs. So I took the protein sequence from P-capsicity and I added a step. I did another protein search or protein blast, and now you can see I narrowed it down to 31 genes. And then you scroll down a little bit, and sure enough, on the second page you can see that I still have my initial gene that I started off with, which made me feel pretty confident that these are in fact orthologs. So now that I know that that's the case, I added another step where I would search just for the domain, just to make sure that all of these contain the DMAP1 domain, which is the gene that I am interested in. So I add a step right here. I use interpro domain, and now it reduces it to 20 genes from my initial 45. However, I would like to point out that there are 2 of ASCIST, so in fact I actually have 19 through FungiDB. Sure enough, as well, you can see I have my original gene that I started off with from Phytophthora and also from P-capsicity. So now that I have the domain, and I am pretty confident that these 19 genes from 19 different specimens are actually present, I proceeded to download all their sequences through FungiDB, a sequence alignment with Clustal. And as you can see right here in the domain, that it is pretty conserved throughout most of it, except for a couple places here or there that actually line up with different, like, so here's albugo, and then here's aphanomycetes. They're a little different, like both of these have a K versus an N. So anyway, I took this and I proceeded to construct a phylogenetic tree to gain an understanding of the route of these genes. And it lines up pretty well. Like you can see Saprolegnia stays together, Aphanomycetes stay together, Halospora, these guys stay together. The Pythium vexins kind of makes sense that's a little over here instead of with the rest of the Pythiums, because its sequence was actually a lot longer than the other ones, so I'm not surprised that one's over here. Alright, so now that I have a phylogenetic tree indicating the evolutionary route, I searched for expression data. To Brett Tyler, he recently updated expression data for Soj after the OMGN conference, and as you can see here, I added a step using his expression data that he uploaded through FungiDB, and you can see that it is actually expressed for Phytophthora Soj, indicating that the gene is there and it has to serve some form of function since it is being expressed. However, I would like to point out one thing to compare later on is that it seems it's more expressed here in mycelium, while in P-capsicate it is a little bit different for the expression levels. We hope to later on do qPCR runs of our own to reaffirm this and just look into it. Alright, so now let's say we want to find expression data for an organism that was in my table, but they didn't have expression data uploaded through FungiDB. Thanks to the OMGN bioinformatics workshop, I knew how to do this, so I started searching for SRA files, which are sequence read archives. So I found one for P-capsicate under 3 levels of expression, one during infection, one during mycelium, and one during spores. So one thing you want to look for is you want to make sure that this is actually expression data. So you want to make sure that this came from messenger RNA. So this says transcriptomatic, indicating that I can actually use this for what I intended. And so you could read into this too and like see what experiment it came from and what they did. So I took my extension number right here and I used it, their sequence read archive, into RNA Rocket or Galaxy. And after a little bit and it uploaded and I had them fragment the sequence, I get this side search over here on this side. And here you can see I end up using a Bigwig, which is a basically a algorithm that will allow me to view data in a graph. There's also other ways you can view it, like you use the expression of a transcription with cufflinks, and then like there's a bed graph where I can basically see where the sequence reads are. Alright, so you can see here I got 3 levels of different stages of P-capsicate where they got expression data. So I took all 3 of these and then I clicked on the FungiDB so I can display the data in their genome browser. So it will take you to this page. One thing you'll want to do is you put your gene number or your gene name in here so you can actually zoom in on the spot where your gene is and see if they have expression data. In my case, I did, and here you can see there's expression of this gene in different stages. As I mentioned before, the mycelium levels are actually lower in this one, while it was higher in excuse me, in Soj. Also, things of interest is that this might be an intron and that this gene model might be a little bit different, since like right here you can see there's expression in what should be an intron. And all right, so now we prove that we have expression data in 2 species for this gene. So we are pretty confident that this actually has a function, and we are proceeding to do the characterization of the gene and try to prove that it actually does undergo methylation. Summary: FungiDB was a valuable tool. It allowed us to prove that this gene is there, that it's expressed, that it might actually have some form of function. If we got no results from our initial bioinformatics experiment or research, then we would have just stopped and moved on to another possible candidate, because if it's not there, why actually waste time and money? So I also like to mention that it's clearly conserved and expressed in oomycetes. We are currently undergoing the characterization of the gene, and we would like to state that we believe oomycetes do in fact undergo methylation. And if you wanted to discuss this in person, I will also be presenting a poster here, so feel free to stop by and talk about it later on if you want. No, you're fine. I would like to acknowledge our lab members, particularly Shannon, who also went to the bioinformatics workshop with me last year. There were a couple of moments where I wanted to rage quit and just stop, and then I kind of went to her and we talked about it, and then I ended up working my way through it and figured out how to actually do most of this bioinformatics stuff. I would also like to thank CURS, which is a Center for Undergraduate Research, and they basically paid me so I could concentrate on research rather than having to actually get a part-time job, along with the Bioinformatics Travel Award last year, because without it I wouldn't have been able to figure out half of the stuff on my own, and I probably would have quit. Um, I'd also like to thank the OMG and Travel Award, which allowed me to go to the conference here like a month or two ago, which I was really appreciated. Actually got to go. I also like to acknowledge Ohio First Bioinformatics Workshop, which is another scholarship I got, which was related to bioinformatics, which helped me along the way. And also, yeah, and that's my talk. Thank you.